Back to yet another journal club brought to you by lifespan.io. I'm Oliver Medvedik, and we have yet another wonderful journal article for everybody here in the aging community. Summer is upon us. It's pretty hot here in New York City. I don't know what your temperature is out there, but it's kind of roasting out there. I've got a fan over my head, so it's not too bad. Let's see, what is the journal article that we have today? Well, we have a journal article on fecal microbiota transfer between aged mice and young mice, and vice versa. And it sort of fits the whole kind of schema of many articles that have been coming out over the past number of years, and also recently, where we're seeing a number of, well, I could say substances, fluids, etc., being transferred between old organisms and young organisms, and young and old, and seeing effects, either positive or detrimental. We've covered papers in the past from the Convoy Lab discussing heterochronic plasma transfer between young mice and old mice and seeing beneficial effects. In this case, it's just a tad bit removed, in that instead of transferring bodily fluids, I guess fluids from the blood system, we are transferring fecal microbiota from young mice to old mice, and seeing what effects, if any, there are. And previous papers have noted positive effects with fecal microbiota transfer, I believe in mice that were progeroid mice, so transfer from young mice to progeroid mice seemed to have a beneficial effect. And also it's been well documented that the microbiome changes as we get older. So the question is there, for normal aging, is this a causative effect? Obviously there's a correlation, but is the change in the microbiome a result of the aging tissues of the organism altering the microbiome, or is the microbiome itself doing some sort of alterations to the tissues of the organism? In this paper, well, it could be still both, but this paper shows quite clearly that in many cases here in the organisms they're looking at, the microbes themselves are playing an active role, or at least the substances that the microbes can secrete are playing an active role in either positively or negatively affecting the physiology of the organisms. So let's share our screen here. So this is our paper. Move this to the side here. Fecal microbiota transfer between young and aged mice reverses hallmarks of the aging gut, eye, and brain. So quite a bit of so obviously, you would expect there to be some effects at the gut level, because this is the microbiome they're looking at. They're looking at the gut microbiome. But you kind of, a little surprising, to see these effects way upstream in the eye and the brain, and seeing really significant changes, neurophysiologic changes in the organisms. I believe this is Parker et al., and this is from the Norwich Medical School, University of East Anglia, Norwich, in the UK. So, in Steve and Fatima's neck of the woods, I think. I don't know how close this university is to you guys. I think you're somewhere around the London area, right? Suburbs of London.
We are not anywhere near London.
Oh, you moved. That's right.
So no, yeah.
Are you by the Norwich Medical School?
Norwich. It's Norwich.
Norwich.
And we don't. No, we don't live anywhere near there. That's right over on the east coast. So I would say best guess, couple 100 mile, maybe 300 mile.
Okay.
We're about as far away from the sea as you can get here, pretty much.
Gotcha. All right, so let's take a look and see what is going on here. So let me zoom this out a little bit, occupy all my space. So this is basically a graphical abstract, a summary of their experiments. So fecal microbiome, so basically mouse poop was being transferred One of two ways. They had the mice basically equilibrate the mice that they had in the study in their own cages, because they're basically surrounded by poop and they eat poop occasionally, so some of it gets naturally transferred, but they also, I believe, transferred it through oral gavage into the mice. So you had a transfer from young to aged mice, and also aged to young. So one thing that was noted is that they observed a negative consequence of having an older microbiome go into the young mice. So they looked at the CNS, they looked at circulating inflammatory factors, and they also looked at the gut lumen and effects thereof. And they also looked at the fecal microbiome composition as it changed. So this is just a summary of what they did. So let's see, let me scroll through here. This is their experimental layout. So they had the young mice were 3-month-old mice. The old mice, they had old mice that were 18-month-old mice called old, and aged, really old mice that were 2 years old, 24 months. And they did a number of different transfers. So the numbers on the left are the numbers of mice. So they didn't have as many aged mice. They had 16. And they basically, I think they broke them down into various grouping sizes. So you see that, 9, 7, 7, 7, 7. So young mice, so, you had young to young transfer. So that's the blue arrow here, pooled young fecal slurry, right? Sounds pretty gross. So basically a fecal smoothie given to young mice, from young mice, and also fecal slurry given from the aged mice to the young mice, and also from the old mice to the young mice, and also control. So they had controls where it was basically either PBS given or only antibiotics. Now, what they did with the mice, the way that they set up the experiment is they cleared out the gut microbiome using a variety of antibiotics. There's a couple of them, I believe 5 of them. 2 of them were given in the water, several through oral gavage because they didn't taste good. What do they have? Vancomycin, metronidazole, haven't heard of that one, ampicillin, neomycin, so 4 antibiotics. And then the mice were put back to their cages with their partners to kind of equilibrate, or they were, if those were the controls, or they were basically after several days of washout, after given the antibiotic dose, they were transferred, the fecal slurries were transferred into the mice. And then after X amount of days post fecal microbial transfer, a little after over 2 weeks, various assays were performed. So immunostaining, looking at the histopathology of the brain, NMR being done on the metabolites in the blood, ELISA assays on protein expression, and so on and so forth. So they tried to control for a lot of variables here. They had a number of mice. I think they also, within the control groups, they mixed them around in the cages to make sure that there was minimized kind of the swing in variation between the microbiomes. And then they performed the analyses. So I'm going to scroll down here and stop sharing for a little while. And the first thing that they did, first experiments that they did, so this is all experimental backgrounds. Is that they looked at the brain. So where's the, right, okay. So they looked at inflammatory markers within the corpus callosum and also the cortex of the mouse brain. So I'm going to pause here for a little while I drink some coffee and kind of let that, a fecal smoothie, oh my God, LOL, yes. That's what it is. I don't know what to say. I'm going to withhold on the jokes. I'm going to filter myself, put the force fields up.
Yeah, that's not going to happen. You know it. you're going to break sooner or later because you did when we did that
Okay.
the microbiome special with Mike Lustgarden and Amy Prow and what was his name now? Cosmo. Yeah, and you lasted 20 minutes before you made the poop joke, so don't feel so bad if you break your record today.
Okay. All right, somewhere in California. Okay, all right, that's it. Oh
dear.
Okay, let's go back to screen sharing. Okay, so what did they look at? So they looked at microglia. So microglia are basically cells in the brain that scavenge. Well, they do a number of things, but one thing that they do is they scavenge damage. And there's markers that could be used to label them. These inflammatory IBA1 plus proteins that are, I believe, they do a number of things, but they correlate with basically migrating microglia. So if you have more of these IBA1 plus microglia, that means that something is amiss. So let's see what they did. So they did immunohistochemistry here, staining. So this is the mouse brain. This is the part of the cortex they looked at, and this is the corpus callosum, the connecting part. So gray matter and white matter, these are basically the cables that connect the two hemispheres of the brain. Hook staining, basically for the cells themselves, the DNA. And let's see what else we have here. Oh, and well, those are the actual cells that are stained. So if you want to take a look at the beautiful qualitative data there, and these were the experiments. So so these are the numbers of cells that were stained per particular field that they looked at microscopically. So here are controls: young PBS, just PBS given to the young cells, that were treated with antibiotic and then allowed to basically go back to their cages, so their gut was repopulated. And then young to young transfer, so that's kind of a real true positive control here. And then you notice now young to old meaning these were 18-month-old, and aged being the really old cells, so the 24-month-old. So you can see there's definitely significantly much more within the cortex IBA1 positive cells, microglial cell activation going up. Now, there's something interesting embedded in here, which the authors actually point out, and I'll get to it in a moment. And they kind of don't look at it for the rest of the figures, and just kind of interesting why. But so here's the baseline for old cells, old PBS. And notice now that if you treat the cells with antibiotic and you clear them out, and then you assay them, so these are really short duration. So I believe this is like 2 weeks afterwards. So this isn't like they haven't looked at this. One caveat for these experiments is that they haven't tracked these mice like many weeks later or many months later, right? So this is within a short window afterwards. So you notice that after antibiotic treatment in the old, it goes down. Now they haven't done the antibiotic treatment on the aged mice, and the reason is that they didn't have that many to work with, the really aged mice, 24 months, so they had to use them for other controls. And old plus young wasn't really that significant. Old aged actually also made it worse for the back to the baseline of old. And then with the really aged mice, when they transferred the young microbiome, you had a decrease in these IBA1 positive cells. And you see similar effects for the corpus callosum. Now I'm going to kind of stop sharing here. What the interesting thing is to me is that you do see negative consequences of having old fecal microbiome being transferred to young cells, and you see a positive effect, at least for the aged cells, when they transfer the young. But you also see a trend. It was kind of on the edge statistically of getting rid of the microbiome in the old cells using antibiotics and not transferring the young cells. And that kind of, to me, hints, and I think the authors mentioned that in a sentence or two in the paper, that, not that there's probably not just a positive effect of a young microbiome effect happening here, but there's probably some negative metabolites or negative effects of an aberrant microbiome that's in an old mouse, right? So to me, that's kind of hinting at that take-home message. Now, interestingly, when we go through the other experiments later in the paper, they don't have that antibiotic-only experiment. So I would be kind of interested to see kind of some of the experiments repeated with that, right? To see whether or not some of the effects that they see in an improvement in the intestinal integrity, which will come up in latter figures, for example. It's not just because of a transfer of a young microbiome to an old intestine, but perhaps just giving high dosage antibiotics and getting rid of some of this, some of the negative microbiome might also have an effect. Yeah, Steve says it's like the convoy's blood filtering. Old blood can rejuvenate too if the bad factors are removed or calibrated. I think that there might be, and one thing to kind of tease apart, I think, is what the contributions are between positive and negative factors, right? So there's probably a pie chart that you can make that says that there's a negative effect of some of these factors, but there's also a positive effect. And what is that percentage? So running experiments like this where you just deplete the microbiome using antibiotics without introducing anything new, so basically having these mice sterile and seeing what improvement there is in certain factors, and then reintroducing a young microbiome and see if you get an enhancement. Perhaps you can determine what percentage, what contribution there is between a negative effect versus a positive effect. So that's just my 2 cents here. Okay, let's go back to the paper. So I believe we have similar effects happening with the IBA1 positive cells and the corpus callosum. So a similar trend also with the old, the p-value is kind of on the edge there of the old mice that are treated with the antibiotic cocktail and not having a fecal microbial transfer take place. So, and the fact that, of course, that if you put aged into young and you see a aged plus young, where's the young plus aged? Young plus old, young plus aged. And of course, if you see an increase in IBA, that also suggests that you have a negative effect here, right? Because you're getting the aged microbiome put in there. So that's a nice kind of nice correlation between the two, I think. Antibiotic treatment of old, get a decrease in IBA1 plus cells, and then transfer after antibiotic treatment of aged microbiome to young mice and have also an increase in IBA1 plus cells. Okay. So next they look at the eye. So they look at the retina, which is quite sensitive to a variety of toxins, obviously. So they look at different components of the retina. They look at, I keep forgetting the name, it starts with a B, it's a membrane. So they look at the retinal pigment epithelium, which is lining the retina, which supports the retinal cells. And then there's a kind of a basement membrane called, I'm going to garble it, Bruch's membrane, where basically that also supports the cells. And they look at various factors that correlate with inflammation in the retina. So they do the same tests, same experiments, but they're doing cross sections and they're staining for complement C3. And here there are a variety of stained backgrounds. So controls, old PBS, young PBS, complement C3, which is in green. So more of it, the more inflammation there is. Aged PBS, so a lot, and then young plus aged fetal microbial transfer, and then aged plus young fetal microbial transfer. So you could just see from these images here that you get basically a rescue or a reversal of complement C3 in the retinal pigment epithelium. So, and this is basically just a quantification of that. So again, one thing I noted in the from the previous figure, we don't have here, so we do see that adding aged fetal microbial transfer, adding the fecal slurry from aged 24-month-old cells does have this negative effect on the retina in young mice, and then you have a reversal if you take aged and transfer the young. So again, suggesting that there's not just positive effects from the young fecal microbiome, but there's probably, well, no doubt, negative effects from the old fecal microbiome as well. And they look at a different area of the retina. So this is, I believe, is this Bruch's membrane? No, this is just C3. And then they look at protein retinal. This is a protein that is positively correlated with the health of the retina. Retinal pigment epithelial protein 65, which is involved in supporting the retinal cells, and it goes down as cells are aged, as retinas age, and it is up pretty high when the cells are young. And you see also this effect where when you add aged fetal microbial transfer, the level of this positive protein goes down, and it goes up when you transfer the aged microbiome to the cells. So, and they look at various cytokines. So basically, these are all correlated. So in aged mice with the young fetal microbial transfer, so these are all correlated with inflammation. Ones that have a positive effect, anti-inflammatory, are high. Ones that have a more of an inflammatory response are lower in proportion to the aged mice that did not have a young fetal microbial transfer take place. So it's pretty interesting that we're seeing these effects pretty far upstream from the gut, both in the eye and in the brain. So you would expect to see, probably if we looked at other organs, other tissues, you would see. This would suggest to me that you would see a lot of these effects recapitulated throughout the animal. At least this is strongly suggesting that. Of course, they didn't look at the kidneys and liver, and et cetera, et cetera. Interestingly, they mentioned in the paper, it's in the supplemental data, that they didn't do any long-term memory tests, I think, like the Morris water maze test, but they did some other short-term tests with the mice, and it, at least for the short term, within like 2 to 4 weeks, within that period of time, the mice did not seem to have any kind of improvement in their functionality. Now that just could mean that the effects are a bit more subtle, and that they need to have an adequate test to basically see if the mice are actually improving cognitively as a consequence of these fecal treatments. Because again, the one other issue is that this is pretty short term. I think they looked at the mice within like 2 to 4 weeks after the fecal microbial transfer. So a pretty short duration, enough to see these physiologic responses, enough to see alterations in protein levels, but probably not enough to actually see what's happening at a cognitive level. Okay, so any questions, any discussions? Seems pretty straightforward. everybody's all quiet.
Oliver, would you consider doing this yourself?
That's a good question. I think with let's say that maybe not at the moment, but I think if looking at kind of all, so short answer is yes with caveats. Longer answer is if we if my fecal microbiome and other microbiomes are aberrant, right, and we basically are capable enough to basically have a pattern that says, okay, you have a bad fecal microbiome, and we have a protocol here that is going to revert it to something normal and healthy, and it works, and the risks are pretty low, then sure. I think these are sort of this is sort of a lot of these potential therapies where you have a transfer of components are fairly low risk, and if there are benefits that can be quantified, such as heterochronic plasma transfer, then yes, sure.
So I have heard of human clinical, I don't know if they're experiments or treatments, where they've done fecal transplants by the enema route rather than by oral gavage, and I wonder how comparable this is.
Yeah, I'm not sure. I mean, obviously, you can do it both ways. One way is more convenient. You could have timed release, right? Capsules that basically release in the lower intestine. I mean, people been taking probiotics for a long time. So I think like I said, the short answer is yes. How long these benefits last, it's questionable, because obviously the rest of your tissues are aging, right? So the environment where these where the microbiome could interact with cells that probably the environment's going to change. I would assume that these effects go down over time, and that that's just an assumption I'm making. So clearly more experiments need to be done to quantify how long this effect lasts, and I'm assuming that there is going to be a drop off. But it seems like given the limitations, at least here we're seeing that there is a benefit. Interestingly, I think in some other neurologic disorders, people have been experimenting. I think there is I don't want to say ALS, but there have been some there's some clinical trials, I think, starting right now with a fecal microbial transfer to kind of repopulate a healthy gut microbiome, to see if this could alleviate certain neurologic disorders that seem to be triggered by inflammatory responses. Having, for example, I do know that in some neurologic disorders, the myelin sheaths are attacked, and complement plays a role. So there might be, there might be a connection there between the gut microbiome and neural health. Well, certainly here they suggest that. So it's very intriguing. So yeah, long answer, but yeah, I think if the data comes in, then sure.
And I see a comment in the chat there.
Yeah, Satova Wolf, great data for resolving C. diff in humans. Yes, C. difficiles. In fact, actually, my sister's a nurse, and that's something that she's been reading up on a lot. So yeah, for C. difficiles, Clostridium difficiles, which is a big thing in patients that are infected have their guts infected, this microbial transfer seems to. Oh, sorry, Michael. Jeez.
I don't mean to interrupt. Please keep talking.
No. Please interrupt.
So, regarding what John asked about, like, would you get this a fecal transplant? Based on this paper, I don't think this paper. So a devil's advocate reading of this paper is that this paper might not provide any evidence one way or the other about whether fecal transplant benefits, and it's all about that missing control. Like, this paper has a lot of really good sorts of analysis, most of which deals with inflammation, but they also look at like permeability and other things. But when they show their antibiotic only control, they recapitulate a lot of their effect.
Yeah.
And a lot of the time they're missing the killer control, which is they're really old mice just with antibiotics, no fecal transplant. And it's totally possible that just is the same or nearly the same as a fecal transplant. And they have a good reason they didn't do it, because it's hard to get aged mice and you can't do every control. But it's, from my perspective, it's kind of the elephant in the room. Like, one possible reading of this paper is that the heterochronic fecal transplant might not have any good effects. It could just be the antibiotics. And except when you take young mice and give them old fecal transplants, okay, that's bad. But it might not even be the bacteria. It could be just like lipopolysaccharide or like leukocytes carrying along in that smoothie they're making. Because there's like the authors of this paper make a point about how independent of, there's two things that happen with age in the gut. Like the composition of the microbes changes. That's a fact. It's also true that gut permeability increases with age. That's also well established. It's not necessarily true that both matter. It might be true that just permeability matters. And if that were true, it could explain away some of the results they're attributing to the changing composition of bacteria. I don't know if that explanation I gave is true, but the real experiment they need to do these antibiotic only controls to figure out, like, does the smoothie really matter?
Right. Yeah. I agree. I mean, because it's kind of, it's you had that response in the first figure, and then they stopped using the antibiotic only for everything else. It is, though, intriguing that that you're getting this negative effect. Again, it could be due to a variety of things, but that you're seeing this negative effect when you get aged microbiome getting into the young. But yes, longer term also, experiments are necessary, because they're seeing a positive effect with clearing out the microbiome, using antibiotics, but that might, again, to kind of like put a kind of a more of a positive spin on this, they might see that go away if they have these mice continuously given antibiotics to get rid of the bad bacteria for a long time, because you need a healthy gut microbiome, and if you keep purging it all out, that positive blip that you see with like less microglial cell activation within like a 2 to 4 week window, will start to probably collapse. I'm predicting, like, if you keep hammering them with antibiotics and then test them 4 months later, 5 months later, then I would predict that that based on how people respond to taking antibiotics for a long time and so on and so forth, that and side effects, etc., that you're going to, unless you repopulate with a healthy microbiome and stop giving these antibiotics, that you probably you'll have this you're seeing this temporary effect, right? And it'll probably become negative. So I'll hypothesize that if they did another follow-up paper to this to include those antibiotic controls, and then let it go longer, and then say, okay, I would think that with the 24 months, I would think if they did all the other experiments, they would see that positive effect. I'm predicting, like you mentioned, Michael, but then it would probably go away after. I mean, if it didn't go away, it would be weird, right? It's like,
right.
Then what do you need a microbiome for in the gut, right? You would probably see other negative consequences months later down the line, and then that would be, blocked, or that those negative consequences would perhaps go away if you stopped giving antibiotics and you basically, gave a young microbiome. So that would be kind of a more solid, right? Like, I think, if you could show that, then it would probably be a more, a solid, data that would suggest that a young microbiome is doing a lot of the heavy lifting.
I mean, if we're talking about our like dream follow-up paper, I would love to see more analysis of the composition of the fecal transplant, and in particular, I would love to see innate immune system composition. Like, are you actually carrying through leukocytes? Are you carrying through a whole ton of IgM? Or like complement, or just like tons of LPS, independent of your bacteria. Because like if you have an inflammatory microenvironment in an aged mouse gut, even if you just forget about all the bacteria in there, you put that in a young mouse, that could be enough to cause a bunch of negative effects. So, yeah.
Mm-hmm. So let's take a look at the rest of the data here. I think they look at, so here they look at serum levels that are proxies for a healthy or not healthy gut. They look at lipopolysaccharide binding protein, which levels go up because LPS, lipopolysaccharide from bacteria, leaks into the bloodstream, the plasma, and you've got this protein that goes up to bind to it. IFABP, I forgot what that was. I think it's a protein that binds to some fatty acid. Yeah, intestinal fatty acid binding protein. Key indicator of microbiota-related epithelial cell damage and disruption, and a surrogate biomarker of intestinal epithelial permeability. So, okay. So all these levels, and also IL-6, inflammatory markers, and tumor necrosis factor. So these are serum and IL-6, that's serum IL-6, and small intestine tumor necrosis factor, that's TNF being expressed in the small intestine. So I think that's like sections of small intestine that were analyzed. Okay, so what do we see here? Young PBS and then young aged FMT, the serum IFABP levels go up. Aged PBS and aged with young FMT, fecal microbial transfer, they go down, right? So you have this, you basically, you have kind of a repetition in you see a similar correlation with IFABP levels and also LPS binding protein, lipopolysaccharide binding protein, as we did with the proteins that were shown in the cortex, corpus callosum, the IBA1 plus, and the proteins in the retina. So you see a similar effect here with proteins that are indicative of things getting into into the serum that don't belong there from the microbiota. Let's see, did they actually have, so like serum IL-6, they didn't really have a significant effect. And tumor necrosis factor, slight effect, but it wasn't too dramatic for tumor necrosis factor. And I don't know what the reasoning was that they gave for IL-6. Elevated in IL-6 in young mice reasoning. Well, okay, there is it's kind of a big range there, but yeah, with age, but not really much of an effect with. So they do. So the p-value p03 here is 0.03 is so there's a bit of an increase in serum IL-6 interleukin 6 levels, but aged PBS and young, they don't see, at least within the time period that they looked at, that much of a dramatic decrease, but more of a negative effect when they transferred the aged fecal microbiome to the young cells. So there are negative gut consequences to getting this fecal microbiome transferred to young, and quite a bit of reversals in some of these proteins that bind to bacterial components that end up in the serum. So I'll scroll here and pause for a little moment. The next kind of 3 figures, they look at the microbial composition, the gut microbiome. And I'm not a gut microbiome expert by any means. So, some of these no doubt are, are associated with a negative environment, some with a positive. And they basically give you a whole list of what is typically found. So there is a different composition in microbes between aged versus young pre-ABX. So you have a different composition of so more of this Turicomonas murus enriched in young, and more of this Prevotella MGM2 that's enriched in aged, right? So you have a different clustering of different types of species and genera. So Clostridium, well, this one found in old. Well, I'm not going to read off all the names here, but you basically get the point. There's a different microbiota composition, and that changes. That changes when you basically do compared pre and post fecal microbial transfer. And the shift basically, corresponds to, as you would expect, not all of them, but some of them actually shift to the younger category when you transfer the young fecal microbiome to the aged, and vice versa, right? So you basically get a you get a shifting of species that you typically see in the young, in the old, if you transfer the young into the old, but not all of them. So it would be intriguing. It's intriguing to me why not all of them take. It could be that maybe some of the components in the, again, going into the kind of yin and yang between the microbiome and the epithelial cells in the gut, it could just be that there might be receptors that are missing in the old gut that won't actually, won't provide a hospitable environment for some of these younger microbes to take. So it would be interesting to see, I think what they looked at here, let me share screen again. They looked at aged and young, but it'd be interesting to see if we looked at more, clearly this is a lot of work, but if we looked at more time points with old mice, for example, mice that are slightly older, middle-aged mice, and then really, really old mice, and see what is, and I don't know if maybe somebody published this paper, to see what the efficiency is of transfer between young and old, right? So if you go young to young, you would expect to see all or most of the transfer taking place. So the composition would be identical to the young. Aged, not all of the young microbes actually will remain in the gut after you check 3 weeks, 4 weeks later. But is that is that an all or nothing phenomena, or does it decrease over time, right? Until and does that correlate with some sort of lack of receptor function in epithelial cells in the gut? Maybe, I'm not sure. So That's just that's something that I was thinking about as I was looking at that data.
So for my money, figure 4B is the most compelling part of this paper. They've got figures earlier in the paper that show really striking findings. They have pretty good data showing functional changes. Well, it's not say functional. Yeah, 4B, the LPB. So they've got other figures that show effects of their fecal transplants. So they show stuff happening in the corpus callosum. They show stuff happening in the retina. 4B is a mechanism, or at least it's a potential mechanism. Like, I'm not an immunologist, but my understanding is that LPS binding protein is associated with like all of the big players for inflammation. Like, it's associated with IL-6, IL-1B, it's associated with TNF, and it goes up when something specific happens, when there's LPS in your blood. And they have a really big effect here, where you get like twofold more of this protein with like under your age conditions than your younger conditions. So it's a big effect, and it's effect that could explain many of their other inflammation related effects. Another cool thing about this protein is there's already other papers out there that show it's aging associated. It's not to the point where it's in any of those like blood-based biomarker clocks, but it probably could be. It's one of those markers that like linearly goes up with age. So being able to revert that, like I feel like I was kind of bad-mouthing the paper earlier, saying, well, maybe the FMT isn't really because of the FMT. On the other hand, if you can get an effect like this, maybe you don't care, because this is like impressive.
Yeah. So again, it'd be interesting to see how long-term this effect is. Because again, that's the other caveat here. Besides the the use of the antibiotics, being used in the first figure, this is pretty short duration. So I wonder how lasting this is. And they looked at only aged here. So it's a pretty robust response, nonetheless, for aged and young FMT. I mean, it almost goes back to young levels. Okay, I think I'm still sharing. Yeah, I am still sharing. So the last figure is they looked at various metabolites, alters lipid and vitamin metabolism following heterochronic FMT. And again, some of these correlate to, positive conditions. Again, there's a lot of metabolic data here between young and aged FMT and aged and young FMT. I believe that various amino acid biosynthetic pathways are improved. I'm trying to find where that is right now. So it's not surprising that the pattern of the biosynthetic pathways that are correlated with various species of bacteria and their metabolites changes, because the composition of the microbiome has radically changed. So and this is going to be shifted. So I don't know which of these off the top of my head are going to be the most useful from an anti-aging perspective. So aged plus young FMT, so improvement in biosynthesis, for example, and a variety of other biosynthetic pathway improvements that you see as a consequence of transferring the microbiome. So does anybody have any comments on this? Yeah. So so there you have it. There are positive effects in the short term with these mice. seeing, we've kind of touched on some of the issues that I think could be taken care of by having see what the effects of having just the antibiotic control alone, and also doing it a little bit long term, right? Because the authors did note that they didn't note any neurologic effects, memory enhancement. But again, these were short-term studies, so it's maybe not too surprising that altering the gut microbiome within 2 weeks is going to have a dramatic shift in memory enhancement in the mice. I would be surprised. So all of this and also previous papers on fecal microbial transfer, I think I think it bodes well, that more research should be done. There's already positive clinical effects with fecal microbial transfer in humans, although not from an aging perspective, but from a perspective of getting rid of bad infections from C. difficiles, for example, infections in the human microbiome. Repopulating the microbiome with a healthy microbiome after depletion through antibiotics or other types of stress that happens to the human gut. So this is a pretty wide field, and this kind of fits within kind of the puzzle of I don't know, just to use kind of a colloquialism, topping off the fluid, so to speak. Right, in somebody that's older. There's a paper that we were thinking about doing, but we'll probably do it next time. It was a recent paper that just came out where they transferred cerebrospinal fluid between young and old mice, and I think they narrowed down the factor that was responsible for an improvement in. I haven't read the paper, so I'm not going to say an improvement in what, but an improvement. And I think one of the factors that they isolated was FGF17, fibroblast growth factor 17. So you've got papers out there showing improvements of transfer between young and old of cerebrospinal fluid, blood serum, and also whole blood, I believe. Also just serum albumin. Getting rid of old serum and replacing it with something that's taking out negative factors, and also now fecal microbial transplantation. I don't know what other transfers that are on the horizon that I can think of. Snot, probably not. Yeah. So. But yeah, it's very intriguing. It's very intriguing that we have a transfer of basically, well, in this case they are cellular components. They're it's the microbiome, but you have components that are outside of your complement of diploid cells, being transferred from young and old and young, and having these very significant physiologic changes occur. Whether or not they're for the long term, or how much of improvement there is for the long term, it's still open for discussion. There's still more experiments that need to be done, but at least for the short term, we can measure these effects, and they're actually happening. So it's pretty intriguing. Yeah. So that's. So it's sort of, to me, that's sort of a complement to, stem cell transplantation or cellular transplantation. So you have a non-diploid cell transplantation of stuff, right? That is having a beneficial effect. Yeah, Sven said it's also interesting in light of their antibiotic results that germ-free mice live slightly longer. I did not know that. Another caveat for this paper is they did this on C57 black 6 mice, and I believe they're only male. I could be wrong. Female mice do tend to also live longer. So I'm wondering whether or not you would get as robust of a response if we did female mice. somebody could double check this, but I think in the materials and methods they only used C57 black 6 male mice, I think. And I'm not sure. I'm sure why. Well, it would obviously give cleaner data because you're controlling for as many variables as possible. But some developmental abnormalities in these mice, like intestine zone development. Yeah, and also of course germ-free mice are always going to be permanently in a germ-free environment, right? So that's kind of the caveat. Having antibiotics blasting through you or having no microbiome is probably not beneficial if you're in a outside environment with everything else. But I didn't know that the germ-free mice live slightly longer. Quite old data from the 70s. Interesting. Okay, so yeah, any thoughts, comments from anybody on this paper? And what other fluids can there possibly be? Cerebrospinal fluid, blood serum, microbiota?
Better stop you right there before this goes too far. So what sort of fluids no, I'm not going there. So yes, I think that's probably an appropriate place to stop. You've been very controlled so far, and let's keep it that way. So you want to talk about, what was it next time?
We're gonna get it from behind the paywall, but yeah, we've got the cerebrospinal fluid paper on FGF 17, and also there's another intriguing paper where, apropos to the open discussion we had on different theories of aging. There's a very recent paper that just came out that basically showed, it was pretty straightforward data. They looked at many different animals, many different species, horses, humans. I don't think they looked at dolphins, but naked mole rats, squirrels, et cetera, and basically showed that the rate of somatic aging, looking at point mutations and other types and insertions and deletions and so on and so forth, very closely correlates with the maximal lifespan of an organism. So they looked at various somatic cells. I think they looked at intestinal, so they looked at just intestinal epithelia from all of these different species, and they plotted it, and it was it was again, lending credence to the model of aging and DNA repair. And I think that was finding one, and finding two was that even though the rates were the rate was of aging was rate of mutation was inversely correlated with the maximal lifespan, the maximal load of somatic cells at the end of that organism's lifespan was sort of similar, within maybe like a factor of 2 to 5 within a wide variety of species. So it's sort of like you can take a certain maximal load and that's it, but the rate at which you get there is different. So obviously the rate being higher for short-lived animals and the rate being slower for long-lived animals, the rate of aging, which would make sense if longer-lived animals have a more efficient system for dealing with DNA repair and other types of mechanisms of repair. So that was I think a nice clean paper that again really ties into the discussions on theories of aging, certainly giving much more credence to the non-programmed aging model there. So
had to get that in there, didn't it?
Yes.
You had to
Anyway,
get that in there. It's probably both.
Yeah. Anyway, so there's so those are the two other papers that I was thinking about. You know what? And in our next journal club, we might do something totally different, because something is going to land in our laps next week, and we're going to be like, oh wait, we got to do this one instead.
Yeah, there is a lot of stuff going on, and you can never tell what's going to happen from one week to the next. Well, you can actually, if you visit our website and look at our various coverages and videos. That will give you an idea of what's going on, and there is a lot going on. It's almost enough stuff to cram into an event or maybe a conference. Oh yeah, that's right.
Oh,
there is a conference, isn't there? Wait,
yes.
There's more. But yes, there is. We are doing a 5th conference. Oliver, you are the MC. You are the Rebel MC, and that is August 11th to 14th. It is a virtual conference, and we are hosting our 5th year of the Ending Age-Related Disease Conference, or ERD as we all call it. There are 2 types of tickets. You can have a paid ticket, which, by the way, today the early bird runs out at the end of today. So if you wanted to get a full ticket, $50 cheaper, you can. If you just visit our lifespan.io website, you can't miss the banner at the top, and that tells you how to get a ticket. But also, I'm going to add that there are free tickets too, for those of you who just want the sort of high-level view and the keynotes. They are free to anyone, and we're going to have a keynote every day, and we've got some interesting people lined up to do that. So we've got all sorts of people, haven't we, Oliver? We've got, I believe Brian Kennedy's come in. George Church is coming, one of your favourites. George Church, he's always interesting. And, of course, he was instrumental in the Human Genome Project back in the 1980s. And that guy just does not seem to want to slow down. He's doing all sorts. He's drastically been influential in reducing the price of gene sequencing. He's been doing all sorts of stuff with CRISPR, hasn't he?
He's trying to bring back the mammoths.
To bring back the mammoths. He's experimenting with developing what we call gene drives to introduce into populations of mosquitoes in the wild, so that they no longer spread things like West Nile virus and malaria, by modifying the mosquito in a way that makes it inhospitable, in the case of malaria, to the protozoa that causes the disease. So the guy's just got loads of stuff, and he's interested in reversing aging too, which, you know, I'm on board with that. So he's coming along. Sounds like someone's got a motorbike in the front room.
Yeah, well, you know, I am in New York City, and I have a window facing the street, and such as it is. I'll unmute myself. I should have muted myself.
It's fine. So, yeah, so that's going on. So do visit the website and check that out. And if you, as I say, if you do want to get a discount on a full ticket, which will get you access to all 4 days of talks, panels, on-demand videos, pop videos. I don't know about the pop video bit, but there's a lot of stuff to get your teeth into there. So check that out.
Say the website name again, please.
It's www.lifespan.io. Just go straight there. It's top of the homepage. And also a shout out to my fellow journalists. If you are part of the media, the press, and you would like to come, get in touch with me, and I can think about giving you a press access. And you can email me. Just come along again to the website, check out the staff page, and get my email. I won't say it on here, and I'll sort you out. So shout out to my journalists. Represent. Oliver, any final comments? The who?
Yes, I was about to say, let's see. Oh, here we go. That is, so if you go to lifespan.io, that is it.
Yeah, just click on that link and that's it. So, yeah, okay, so I guess that's about it. We'll wrap it up. Thanks to everybody that's joined us today. And a big thanks to all the heroes who are, I can't speak today, who are supporting us to make episodes like this and all the other wonderful things that we do, all the news, all the videos on YouTube, all our various channels. Your help goes a long way in getting that done. And that's it. You're all helping us take the fight to aging, and maybe we can win. We'll see. Sure, I'll write about it if we do.
Alrighty. Well, thanks, everybody.
We'll see you next time.